So today I'm going to talk about community ecology and diversity measures. So first of all, there's lots of ways to define communities. Communities can be a group of interacting species that occur together in the same place and time, and they can be a mix of species or they can be subsets of species. So for example, you can define a community at whatever scale you're interested in. So the scientist gets to decide what is the community to some degree. Or it could just be a subset of all the species present. So you could be looking at just the bird community or a particular guild. So like, for example, the picture in the top right-hand corner is showing you all of the nectar feeding organisms. You could also look at a functional group, so nitrogen fixing plants, or you might be interested in just a specific trophic level, like say predators or omnivores. So before we get into this, I just want to kind of double check that you remember some of your basic species interactions. So maybe pause the video and try to fill out this table. Great. So now that you're back, how did you do? These are some of the ways a species can interact with one another. So when you're thinking about positive interactions, you're talking about mutualisms. Competition is a negative interaction. We'll go into a lot of detail about competition. Commensalism and amensalism are a little weird and probably the ones that you have the hardest time remembering. Commensalism is a positive neutral effect, and amensalism is a negative neutral effect. And then things like allelopathy, herbivory, predation, and parasitism are all positive for one organism and negative interaction for the other. Okay, so now that we've kind of thought about those types of interactions, you can start to put them together into what are called interaction webs. And so here you can see an interaction web diagram. It's more complicated than a food web. It's similar to a food web, but it includes other things, these horizontal interactions other than just trophic interactions. So you still have the trophic interactions, which are kind of shown by the steps up through the diagram. So from primary producers to primary consumers and secondary consumers, all the way up to tertiary consumers. Now you've got these kind of side-by-side interactions that show you something about competition or some kind of a mutualism or these other interactions. And so they're more complex than food webs, and they show things other than predation. So a lot of what we're going to talk about today is how communities are structured. And structure is a really interesting way to think about communities. It has to do with how they're put together, who the components are, how they function, how these different species interact, and how we can start to kind of make measurements of community dynamics. So how is the community put together? First of all, you could think about species richness. So richness is the number of species present in a community. In both community A and B, there are 4 different species, but you can see the communities look really different. They're structured differently. Right? So, but they're not different in terms of species richness. Now, they are different in terms of species evenness. So what you see in community A is that the bulk of the individuals are these kind of bluish, blackish butterflies. Okay, so individuals are not evenly distributed among the 4 species like they are in community B. Community B is a much more evenly distributed community. And together, richness and evenness tell us something about diversity overall. And so we're going to figure out how to measure diversity in this week's lectures. And we're also going to try to understand a little bit about species composition. So who is there and what do they do? So here we're going to walk through real briefly Shannon's diversity index. There's a Chalk Talk lecture that walks you through how to do these calculations. But basically Shannon's diversity index is H or H prime. And it's basically the negative or the inverse of the sum of each species, the proportion of individuals found in each species times the natural log of that proportion. So if you have S number of species, right? So S stands for richness in the community, then you have I from 1 to S species in the community. And this is a little bit tricky. It's summation notation, but it basically means that for every species in your community, you're going to figure out the proportion of individuals that make up that species. And then you're going to multiply that by the natural log of the proportion. You're going to add those all up for every single species in your community, and that gives you age. And again, this is probably not the best way to explain it, but the chalk talk goes through all the details. So see that associated chalk talk. So here you can see just two tables, again, corresponding to community A and B, and they show you how you would calculate those different proportions and multiply by the natural log of the proportion and then add it all up. And what you can see is that for community A, species diversity is much lower than for community B. And you can see that because of that Shannon's H value, which is 0.5 for community A and 1.3 for community B. And it looks like in the book, it's not taking the inverse of Shannon. So you see some slight changes to these indices when you're looking at them. And so I were reporting the Shannon's diversity index value. I would report 0.5, not negative 0.5, because I like to take the inverse of that. Okay, so diversity and biodiversity can be described at all these different scales. So I've been mainly talking about species level diversity, but there's trophic diversity, there's functional diversity, there's genetic diversity, there's, kind of regional scale diversity as well. And so biodiversity studies can go, from the tiniest thing like the gene all the way up to the landscape. So you can kind of think about species in a community like jelly beans in a jar. Okay, so what you can see here is that we've got lots of jelly beans of lots of different colors. Each color represents a different species. Okay, so imagine all these different individuals. You can see it's pretty well mixed community in this jar of jelly beans. We could sort these beans by color, okay, and then we could count up how many of each color. That would be the abundance for each species in the jar. And of course, the number of different colors in the jar is your species richness. Now, what if there are some really weird colors or flavors hiding in the middle of the jar, but there's only one or two? Those would be rare species in your jar, right? And so we have to take into account that when we're sampling from this jelly bean jar of diversity, we might be missing some really rare species. And so I'm going to talk about some ways to deal with that. Okay, so unfortunately, sampling species out there in the real world is much more difficult than just counting jelly beans. But imagine there's this unknowable kind of jar of jelly beans that represents all of the individuals of all of the different species in your community. And you're sampling, you're grabbing a handful of jelly beans, okay? So those really rare species, you might miss them when you grab them. And that's true for sampling. If you're sampling aquatic insects or birds or soil microbes or whatever you're sampling, you're going to miss some species out there. And so any sample, is going to miss diversity. And so we have to take that into consideration. So what we can use are called species accumulation curves to estimate the total number of species in a community. And so we plot the number of species we find as we increase our sampling effort. So for example, you see a table here, and you can see I've got just the individual that's encountered in order from 1 through 9. The first individual I encountered is from species A, and I saw one of them. Then the second individual I encountered was species B, and I saw one of them too, but now the third column tells you how many species you've encountered. Okay, so I saw species A, great. I saw species B, now I'm up to 2 species encountered. The third individual I encountered is another species B, so I'm still at 2 species. And then by the fourth individual, now I've got species C. Okay, so now I've encountered 3 species. And so I've graphed this number of species encountered as I've increased my sampling effort in the blue line. Now, it could have been that I encountered these species in a different order. And so I can take the same data set, and if I shuffle the species column, then I might get 3 different curves. And so I just shuffled these A's, B's, and C's, and I plotted the kind of encounter curves. And what you can see is they look kind of different, right? I'm encountering species in different ways, but they all start at 1 and they all end at 5. Okay, so they're all topping out at my richness equals 5. Now, the curve is kind of starting to kind of like bend, right? And that's good. That means that, I've captured all the species that there are to capture. We can use computer programs like PCORD, which you'll learn how to use in the stats section of this class much later in the quarter. And we can shuffle our data sets like thousands of times to create these smooth curves that show us the overall accumulation pattern. And what's nice about this is it tells us if we're starting to curve and hit that asymptote or not, because that's where we want to be. We want to be capturing all of the species that are possible to capture. So species accumulation curves are also called species richness curves, collectors curves, or species effort curves. And they're plotting richness as a function of the total number of individuals counted or the total number of plots counted. And the goal is to reach an asymptote where you're no longer finding new species. Here you can see a figure from the book which compares these accumulation curves for different habitats. And so the curves are starting to reach their asymptote for the temperate forests and tropical birds and the red and blue dots. But that's not true for human oral bacteria, tropical moths, and it's definitely not true for tropical soil bacteria. All of those curves are still increasing very steadily. So you can use a curve with your own data to see if you've sampled enough. Am I getting to the point where I'm starting to get that asymptote or not? And then you can also predict your actual species richness in a system using a curve fitting model. So you can extrapolate past the data set that you've collected to see what species richness should be if you can estimate it. And so we can use these types of curves really well when we're doing ecological work. And there's something that's really similar to a species accumulation curve, but a little bit different, which is called a rarefaction curve. And a rarefaction curve, sorry, I forgot to put the animation on this slide. A rarefaction curve helps you compare apples to apples. So say you have two communities. In the first one, you find 243 individuals of 31 species. And in the second community, you find 63 individuals, but of only 9 species. Is it okay to say that community number 1 has a higher species richness? What if you just didn't sample enough of community number 2? So rarefaction puts these two communities on equal footing. You put all of your number 1 individuals into a hat, and then you just pull out the first 63 of them, and that puts them on the same footing as community 2. Now, if you do this 1,000 times, I can give you an estimate of the species richness with 95% confidence. And so this is called rarefaction. So say we do this 1,000 times, and we get this estimate of 19 species for community number 1. With a 95% confidence interval of 16 to 24 species. We're nowhere near the 9 species that we got from community number 2, so we can be pretty confident that community number 1 actually does have higher richness, even though our sampling efforts were different. So that's what rarefaction curves allow us to do, is kind of put communities on equal footing. And there are some assumptions of rarefaction. 1, communities are closed, there's no migration. 2, sampling was sufficient in both communities. 3, individuals were sampled using identical methods. 4, individuals were distributed randomly across space. 5, individuals were sampled independently and randomly. And 6, the samples are taxonomically similar. And then there's one more thing we can do with these kinds of data. We can do what's called the Chow index. We can estimate the actual species richness. And all we need to know is how many singletons and doubletons are in the data set. And so Chow is equal to estimated richness. And to calculate it, you take the richness that you observe, and then you add this kind of factor to it, the Chow factor, basically. It's the number of singletons in your data set, how many species are represented by just one individual, squared, divided by 2 times the number of doubletons. And the doubleton is a species that is represented by just 2 individuals in your sample. So the Chow index is a pretty cool way to estimate species richness. All right, so take a 10-minute break. We'll finish up the chapter in part 2.